Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled New Options for Avoiding a Dedicated TQT Study, the Impact of the New ICH E14 S7B Revisions. My name is Sonia Hunt, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario delivers the leading endpoint technology solutions for clinical trials. Through experience gained from over 19,000 clinical trials delivered in support of 870 regulatory approvals, Clario fuses scientific expertise and global scale into the broadest endpoint technology platform to enable pharmaceutical, biotech, and medical device partners to transform lives. Through Trial Anywhere, Clario has mastered the ability to generate rich evidence across all trial models, decentralized, hybrid, and site-based clinical trials. With 30 facilities in 9 countries across North America, Europe, and Asia Pacific, Clario's global team of science, technology, and operational experts has been delivering the richest clinical evidence for nearly 50 years. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. Boye Darpo. He is the Chief Scientific Officer, Cardiac Safety at Clario, with board certification in cardiology and internal medicine. He completed his cardiology training at Sahlgrenska University Hospital in Gothenburg and was appointed Associate Professor of Cardiology at Karolinska Hospital in Stockholm in 2000. Dr. Darpo held various positions within CROs and the pharmaceutical industry, covering a wide variety of therapeutic areas. Member of the ICH E14 Expert and Implementation Group between 2000 and 2008, he led the IQ-CSRC study, which resulted in the 2015 revision of E14, allowing concentration QTc analysis to exclude small effects. Now it's my pleasure to introduce you to your second speaker, and he is Dr. Robert Kleiman. He is the Chief Medical Officer at Clario, formerly ERT, and has performed research in both basic cellular electrophysiology as well as clinical cardiac electrophysiology. He practiced clinical cardiac electrophysiology prior to joining Clario in 2003 and has 30 years of experience in cardiac safety during the drug development process. Now it's my pleasure to pass the mic and the controls over to our first speaker, Dr. Darpo. So Dr. Darpo, when you're ready, you may begin.
Thank you so much, Sonia. Thanks for the nice introduction. It's my pleasure today to present on new options to replace the therapeutic study. And my focus, the emphasis here will be on the clinical conduct, the clinical part of how to achieve this. So until recently, non-clinical assays have been used mostly or merely to ensure that a new chemical entity can be taken safely into first-in-human trials. The revised S7B E14 Q&A document from February this year expands this role of non-clinical assays by also allowing results from those from the HERG assay and the in vivo assay specifically to be used to support clinical data. And we can look at this as 2 different pathways. Path 5.1 is not new, but it's further promoted by this most recent revision. So when concentration QTc analysis is applied to data from, as an example, the first-in-human study, And we would like to use that to waive the request for a formal therapeutic study. Achieved concentrations must exceed therapeutic concentrations. It used to be the case, or at least since 2015, it used to be the case that we need to be twofold above exposure levels or plasma concentrations that could be seen in patients. Now, with the revised S7B E14 document, we are by combining negative non-clinical assays with a negative clinical demonstration of absence of an effect in the clinical study, we can reduce that margin. So if we cover what would be seen in patients and then also have negative non-clinical assays, that is the same as to say that we have a negative QT assessment. 6.1 is mostly about oncology. That is when a drug cannot be safely given in sufficiently high doses to healthy subjects. And if we run a study like this in cancer patients without placebo and at the highest recommended dose, and if we, based on that study, conclude that a small effect, so an effect above 10 milliseconds, can be excluded, then the sponsor is allowed to claim a low pro-rhythmic liability if the clinical assessment is supported by negative standardized non-clinical assays. So by 2015 revision of the E14 document, the regulatory scene really changed. That revision was based much on regulators' accumulated experience from concentration QTC analysis applied to therapeutic studies, but also based on the results of the ICH study, which was performed by industry, FDA, and iCardiac in collaboration, and in which it was demonstrated that concentration QTC analysis can be used to detect mild QTC prolongation by using concentration QTC analysis. It's fair to say that today, the majority of therapeutic studies, so those remaining, are performed with concentration QTC analysis as the primary statistical approach, which dramatically reduces the needed sample size. But more importantly, we can apply this to many first-in-human studies by implementing procedures that allow us to extract serial ECGs paired with PK sampling. We typically recommend implementing those procedures in both SAD and the MAD portion of the first-in-human study, because you never know how much of accumulation you will see of the drug or of the metabolites, and then make a decision when and what to analyze based on observed PK, but also, of course, based on project viability. This, in our view, constitutes a very efficient approach for ECG evaluation. So this is how the 2015 revision of 5.1 looks like. If a sufficiently high multiple of clinically relevant concentrations are achieved, then a separate positive control is not necessary, which means that we can do this in first-in-human studies where we never have a separate positive control. And the decision making is pretty much the same as we used in the ICH study. If you are able to demonstrate that your drug doesn't cause anything concerning, so if you can exclude an effect above 10 milliseconds at clinically relevant concentrations, then that is the same as to say that you have a negative QT assessment. So it replaces a negative thorough QT study. And this is what has happened since 2015. If we look at the light blue bars, we can see the proportion of thorough QT studies, the proportion of studies intended to demonstrate that a drug doesn't have an effect. So these are based on FDA numbers, and FDA's IRT are looking at, on average, 55 studies per year. We can see that the light blue bars representing the thorough QT studies have been steadily declining since 2016, whereas the black bars, which is the 5.1 pathway, so concentration QTC analysis applied to first-in-human studies, have been steadily increasing, and is almost now close to 40% of these studies, whereas the oncology studies, the path 6.1 pathway, has remained quite stable. So we can already now see a clear tendency of a reduction of the proportion of therapy studies. And we believe that based on this most recent revision, that tendency will be further strengthened. I share with you two examples from the ICH study, and the first one is moxifloxacin. The graph shows you the concentration QTC relationship. So we're looking at the placebo-corrected change from baseline QTCF on the y-axis and moxifloxacin plasma concentrations on the x-axis. And as you can see, the model predicted effect, which is the black line and the gray shaded area, clearly shows that moxifloxacin, by increasing concentrations, will cause QTC prolongation. And if we, as an example, look at the Cmax of a 400 milligram dose, around 2,000 nanograms per mil, we can see that the effect is clearly above 10 milliseconds. The mean effect is around 14 milliseconds, and the upper bound is, of course, larger. And the corresponding graph for levocetracine, the negative drug, looks like this. Somewhat wider confidence intervals, but we see a very shallow slope of the concentration QTC relationship. We can also see that throughout the range of observed levocetracine concentrations exceeding 1,000 nanograms per mil, we can exclude an effect above 10 milliseconds by looking at the upper bound of the confidence interval. So the gray shaded area is entirely below the 10 millisecond threshold. So what will it then take to obtain a therapeutic study waiver? And I'll just walk you through some of the terminology used in the revised I CH E14 document. And this is the same as been the case over the last years, but now the terminology is harmonized, which is good. So a therapeutic dose is the highest recommended dose in phase 3 and therefore in the label. The clinical exposure, by that we mean Cmax levels at steady state with the highest recommended dose. So the Cmax at steady state in the average patient. The high clinical exposure, on the other hand, is what we used to call the worst case scenario. So that is the Cmax at steady state achieved when the maximum therapeutic dose is given in the presence of intrinsic or extrinsic factors that has the largest effect of increasing Cmax. So as an example, if a drug is a 3A4 substrate and you give the highest dose with a potent 3A4 inhibitor, that is the mean Cmax you need to target or you need to look at to define the high clinical exposure. Please bear in mind also that this applies both to parent drug and metabolites. I'll share with you a few examples, and these are all published, and you can see the reference down at the bottom. The first case is a phosphodiesterase inhibitor in development for treatment of dementia. And non-clinically, the HERG assay was somewhat negative, 4 micromolar, and the heart rate effect and QTC effect was seen, but only at high doses in cynomolgus monkeys. And the data we're looking at here comes from a SAD and MAD study in non-Japanese and Japanese subjects. On the left side, we have a graph over the concentrations in the MAD part. So we can see that we can see a substantial accumulation from day 1 to day 14. If you focus on the two blue lines on top, you see that we have almost a 3-fold accumulation of parent drug. On the right side, we have the we have the change from baseline QTC, with the black line being the placebo response on day 1 and on day 14. And if we focus on the two highest dose groups, we can see that the pattern that we see here does not suggest that this drug has an effect on the QTC interval. Obviously, we're looking at small dose groups. This is a first-in-human study, so typically we're looking at 6 on active, 2 on placebo, and therefore many of these confidence intervals are above 10 milliseconds. But the pattern of mean effect does not suggest that the drug has an effect on the QTC interval. And when we take this into concentration QTC modeling, this is how it looks. So on the left side, you have a scatter plot with the red dots being Japanese subjects and blue dots being non-Japanese. And we can see that we have a relatively good representation of high concentrations above 2,000 nanograms per ml. On the right side, we have a goodness of fit plot, with the black line again, the model predicted effect, and the gray shaded area being the confidence interval. And the red bars are the actually observed placebo-corrected change from baseline QTC, so we can see that the model captures the observed data in an accurate way. We can also see that up to very high concentrations, we can exclude that the drug has an effect exceeding 10 milliseconds. So this is clearly a negative QT assessment. Now for this sponsor, the high clinical exposure scenario was well defined. So with food and with 3A4 inhibition, the geometric Cmax at steady state will reach around 1300 nanograms per ml. So we are threefold above the high clinical exposure scenario, which means that we fulfill the requirements also based on the 2015 revision, and there is no need to reperform or further look into the non-clinical assays. So this is a solidly negative example. The next example is a compound in development for treatment of a CNS disorder, and we have plasma concentrations in the left panel and QTC effect in the right panel. And this is then based on the first human study again in the SAD part, and we can see that the concentrations the peak looks pretty linear, I would say. If we then look at the right bar, we can see that we may have a small effect with the highest dose, the 200 milligram dose group, the yellow line and bars as compared to placebo, but nothing dramatic. And then again, we take this into concentration QTC analysis, and we have the scatter plot on the left side. We can see that we have not so many data points above 400 nanograms per ml, but a reasonable spread at least above 300. And the goodness of fit plot shows you that this drug is negative up to concentrations exceeding, almost reaching 600 nanograms per ml. Now for this project, which we also discussed in this publication, the high clinical exposure scenario is not yet sufficiently characterized. So then the sponsor has two options. One is to perform an additional study in case the high clinical exposure scenario is not covered by twofold. Then the sponsor has a choice of two. Either reperforms a standardized S7B assay, so a standardized HERG performed or conducted at physiological temperature, and in vivo study in dogs or in monkeys to ensure that the drug is negative in the way that Dr. Kleiman would describe later in this webinar. Or you can add higher doses to make sure that achieved concentrations exceed the high clinical exposure scenario. And this is to summarize the revised 5.1. A separate positive control is not needed if you achieve a twofold higher exposure as compared to the high clinical exposure. If this is not met, but you have a clearly negative non-clinical assays, then you can still claim that the drug is negative if the non-clinical assays meet the standards that Dr. Kleiman would describe. 6.1 is mostly about oncology. These drugs cannot be given, many of these drugs cannot be given at high doses to healthy subjects. And also, it is often not justified, ethically justified, to expose cancer patients to placebo. So many of these studies are done with a maximal tolerated dose, the MTD dose, in cancer patients and without placebo. So in these instances, non-clinical assays can also be used for decision making. And importantly, all the necessary components of ECG assessments that we use in first-in-human studies or in therapy studies should be implemented in these studies as carefully as you can, as to the level of quality control as much as you can. And when a sponsor then performs this type of assessment and can demonstrate that with high quality ECGs that an effect exceeding 10 milliseconds can be excluded, this can then be supported by negative non-clinical assays, a standardized HERG and a standardized in vivo study. And if that is the case, and these non-clinical assays are also negative, then the sponsor can argue that this drug has a low likelihood of pro-arrhythmic effect. Obviously, there should also be a negative or a cardiovascular safety database that does not suggest that you have an increased rate of adverse events that would signal potential ECG effects. And also, it should be noted that there is an inserted statement in the guidance that an argument can still be made for a drug that has an upper bound above, somewhat above 10 milliseconds. So that is language that was inserted at a late stage into the document. But basically, your ECG assessment in the clinical study should demonstrate that the drug doesn't have an effect above 10 milliseconds. And this is, I think, this is a repeat what I've already said, I'm sorry, this is the paradigm as it looks today. If you don't have these non-clinical standardized non-clinical assays, then you can still make an argument that the treatment is unlikely to have an actual effect, mean effect larger than 20 milliseconds. This is exactly how the labels look today when sponsors have done, they typically have quite old non-clinical assays and they've run the studying cancer patients and shown that there is no effect of concern, then this is the label that they will obtain, at least in the United States. And this is just an example then of a QT study in oncology patients. So this is a phase 1 study in cancer patients with solid tumors. The data comes from the expansion phase with the MTD dose. As defined in the escalation phase. We're looking at an open label study with 18 subjects, ECGs at baseline on day 1 and on day 15 in cycle 1. And we can see, and then we incorporate serial ECGs paired with PK, and we perform concentration QTC analysis. And we can see that this drug would not cause anything concerning up to quite high or up to high plasma concentrations. So if the predicted QTC effect at the high clinical exposure scenario is less than 10 milliseconds, then if you support the clinical assessment with standardized non-clinical assays, then you can claim that the drug is the drug has a low pro-arrhythmic effect. If you don't have these standardized non-clinical assays, the label will state something along the lines that it is unlikely that the drug has an effect exceeding 20 milliseconds. So why does then precision of ECG measurements, why does it matter? And then we need to bear in mind that we're looking at the confidence intervals. So this is again the levocetracine data from the ICH study. Bear in mind that we're looking at the upper bound, and the more precise assessment you have, the more narrow confidence interval you will see, meaning that you're more likely to exclude a small effect with a highly precise ECG methodology. This graph outlines the main differences between a standard semi-automated ECG method and the EQT method that Clario uses in all studies in healthy subjects. We also have the standard method, and we always use that in patient studies because the level of oversight by cardiologists is somewhat higher with the standard technique. So with the standard technique, we typically extract 3 ECGs at each time point and then look at 3 beats from each ECGs. So on average, we're looking at 9 beats per time point in a process that is highly overseen by a cardiologist. This is a very robust technology. It's been in place for more than 20 years, and it's very useful in patient studies. With EQT, we extract up to 10 ECGs per time point. And all these beats, not only 3 per strip, but all beats are looked at by the underlying algorithm and categorized into high confidence or not so high confidence. The good beats, the high confidence beats, are accepted. The measurement of those are accepted. The measurements are made then by the underlying algorithm, whereas the problematic beats, the not high confidence beats, are looked at by the technician, but there is never any form of adjustment of measurement points. Either the beat is accepted or rejected. So to summarize, the standard technique looks at less data in a process where a technician is allowed to adjust the measurement points. It's a robust method that we use in patient studies, but it also comes with a known variability. In EPQT, we look at much more data in a process that is driven by the underlying algorithm. And this then translates into precision. These are 7 studies that Novartis performed and which we published on some years ago. And the upper 2 graphs shows you the crossover studies, whereas the lower 2 graphs shows you 2 parallel design studies. These are all surrogate studies performed at the same site with the same study procedures. The black lines is the variance in the by time point model using EPQT, whereas the red lines is a standard semi-automated method. And whether we look at test drug versus placebo or MOXIE versus placebo, and whether we look at crossover design studies or parallel design studies, the same pattern is seen. By using EPQT, we're roughly half the variability seen with the standard semi-automated method. And these were all studies, of course, in healthy subjects. So based on this type of data, we always use EPQT in studies in healthy subjects. And this then has a major impact on the ability to exclude small effects. So this is a simulation exercise that we did both internally, published, and then in collaboration with FDA. So if we, as an example, assume a small underlying effect of the drug of 3 milliseconds, which is a standard assumption, and we use EPQT with a standard deviation of around 6 milliseconds, we need 9 subjects on active and 6 on placebo to exclude that the drug has an effect larger than 10 milliseconds. Whereas if we use a standard technique in the same population of healthy subjects, we need roughly twice as many subjects on active and 6 on placebo. So it really matters when it comes to excluding small effects. And my conclusions, based both on the 2015 revision, which allowed us to use concentration QTc analysis, and this more recent revision that will further promote the use of concentration QTc analysis applied to first-in-human study. My conclusions are that early QT assessment performed in first-in-human studies are now a fully accepted way to confidently exclude that the drug has a relevant effect. And this is an accepted approach in all regulatory jurisdictions. I believe, and we believe, that the revised S7B E14 Q&A will further promote the use of early clinical studies to obviate and to waive the request for a later surrogate study. I don't think all studies will all surrogate studies will disappear. There will be options and sponsors that select to conduct a surrogate study, or there will be cases where it's very difficult to achieve sufficiently high concentrations. In those studies, the presence of positive control brings down the need for very high concentrations. But I also think it's fair to say that overall, QT assessment and ECG assessment has been done more efficiently, both by using highly precise techniques and by implementing concentration QTc analysis in early clinical pharmacology studies. Thank you so much. And thereby, over to you, Dr. Kleiman.
Thanks, Borje. Hi, folks. This is Bob Kleiman. I'm a clinical cardiac electrophysiologist, but I'm going to talk to you today about the non-clinical assessment of cardiovascular safety. And this is what the ICH E14 Q&A addresses. And this is really very important. just to give you a little bit of the history, the first version of S7A came out in 2002, and then S7B was released in 2005, the same time as the first version of the E14 guidance. And between 2005 and 2020, regulatory silence, no Q&As at all for S7B. Though, as I'm sure you can imagine, the science has advanced greatly. So this Q&A, just released this year, marks the very first update to S7B. Now, does anyone here remember the SIPA initiative? We haven't talked about that recently, but that's stands for the Comprehensive In Vitro Proarrhythmia Assay. This was first proposed in around 2012 or so. And the proposal was to perform more extensive ion channel patch clamp studies, also assess your drug in human stem cell derived cardiomyocytes, and then take that set of data, put it into an in silico computer model, and predict the risk of proarrhythmia with your new drug. There'd be some verification with human ECGs. But the intent was to replace the thorough QT study with more mechanistic assessment of proarrhythmia risk. Now, despite a great deal of work, this never really met complete regulatory acceptance. But SIPA's legacy is still here. The work on the SIPA initiative was not wasted, and this concept that we can use non-clinical cardiac safety data to complement clinical QT data and therefore perform an integrated risk of proarrhythmic risk is what prompted the ICH working group's efforts that led over a period of several years to the new updates. And as a result, these new updates really increase the importance of the non-clinical data in offering us further options for avoiding dedicated thorough QT studies. So S7B is really aimed at safety pharmacologists. Why does this matter to clinical drug developers? Well, prior to this recent release, the non-clinical cardiac safety data were really important to safety pharmacologists for some decisions they would make before a drug reached first-in-human studies, and then to some extent might help determine the starting dose or understand the ECG monitoring requirements or the maximum dose to be used in phase 1 studies. And the data weren't really used in the regulatory assessment and decisions about the drug at the time of registration. But now, these non-clinical data have become much more important both to regulators and to drug developers in the clinical side. And that means that we're going to have to understand how these trials are done, and we're going to need to be able to review reports from these studies if we need to determine if there's a double negative non-clinical package. As stated earlier by Dr. Darpo, there are two main components to the non-clinical evaluation. First is hERG patch clamp studies of parent and metabolites. And you may also want to do studies of additional cardiac ion channels or other complementary studies. And the second is the in vivo cardiovascular safety study, again of parent and metabolites. And this is a study that looks at the ECG as well as hemodynamic effects of a range of exposures of the drug, which hopefully cover the expected clinical exposure and hopefully exposures that exceed the high clinical exposure. So first, a little bit about the hERG studies. The guidance talks about the hERG safety margin, which is the hERG IC50 that you get from your patch clamp studies divided by the free concentration at steady state for the high clinical exposure. Goes on to explain that it's important to look at the free concentration. That's what ion channels are going to see. So you need to know what human protein binding is. There's some recommendations about how to use that data. And the safety margins should be compared to the range of safety margins computed under the same experimental protocol for a series of reference drugs that have known clinical torsade risk. So generally, each lab should be looking at their historical data on known QT prolonging drugs to establish their safety margin. Although, alternatively, one can reuse a safety margin threshold published by others. Unfortunately, one thing that the guidance and training documents don't really do is give us a single numerical value for what is an acceptable hERG safety margin. Based on the literature, we think that it's probably in the range of 30 to at most 50. A few best practices for ion channel studies. These are the things that you need to look for when you read the report from the ion channel work. First, temperature is critical. The studies should be done at physiologic temperature, not at room temperature, because temperature does affect QTC response. Next, the lab has to have utilized the proper stimulation protocols, and the FDA has even published their recommendations about what that should look like. There should be an assessment of full range of exposures that cover the concentration inhibition curve, not just a statement of the IC50 value. It's also important that they measured bath concentrations of the drug. Some drugs end up binding to the plastic of the infusion setup. And if you don't measure bath concentration, you can't really be sure that what you're testing is what you think you're testing. And of course, you must monitor the viability of the cells. These are isolated cells, and we've impaled them with a very sharp microelectrode. Their viability deteriorates over time, and you have to make sure that you're getting results from viable cells. As well, you want to make sure that both positive and negative controls are tested in the same assay to make sure, again, that the results are going to be trustworthy. And then there's the in vivo cardiovascular safety study. And drug developers and regulators are going to have to be able to look at these reports, which often are performed many years before the point at which we're going to be looking at them, and figure out if they use best practices and if they had adequate sensitivity to exclude a QTC increase that would translate into something that's clinically significant for humans. So we have to understand the design and conduct of the studies and have to be able to find the details we need in the reports. And I've got to tell you, the design and conduct of in vivo cardiovascular safety studies are fantastically different than what we're accustomed to for clinical ECG trials. I'm going to skip over the next couple of slides. They go through a lot of details about how the two types of studies are different. And these are available if anyone would like to look at them in the slide set that we'll make available at the end. But just to summarize some big differences between in vivo and clinical studies. First, size. Typically, an in vivo safety study involves 4 animals, sometimes as many as 8, but typically 4, which I found astounding. In a thorough QT study, even with concentration effect modeling, we need about 30 subjects. Yet here they use only 4 animals. The ECGs also aren't collected as 12 leads. Instead, they're collected via implanted single lead telemetry, typically a lead that represents lead 2. And the measurements are done in a sort of semi-automated method, but they're not measurements of single beats or single ECGs. They're data averaged over a wider interval of 15 minutes to as long as 4 hours. So again, different than what we're used to. They also can't always perform baseline correction and can't look at change from baseline because it's really hard to get baseline ECG data in the animals on the day that they're undergoing testing. That animals are apprehensive, have just woken up and eaten, and their heart rates and autonomic tones are not stable. The PK assessment is different. Again, in animals, you can't collect time-matched PK and ECGs. You can't ask the animals to lie down supine, resting comfortably for 10 minutes. And when you try and draw blood from them, their heart rate and autonomic tone goes through the roof. There are some options to let you make sure that you're studying the exposure range that you think you are. And there are ways to get data that will allow you to do concentration and effect modeling. But we're not going to be getting data from simultaneous PK and ECGs. The animals are freely moving. The assay sensitivity does not need to be studied for each trial, in contrast to a thorough QT study where we've had to do positive control to demonstrate assay sensitivity in every single thorough QT study that we do. And the statistical analysis is different. One difference, as I mentioned, is they don't do baseline subtraction in all cases because it can be very difficult. So they use a single delta comparison to vehicle, which is similar to our placebo control. And apologies, I've got to give you a quick primer on statistics. And I'm a clinical cardiac electrophysiologist. Me giving you a lecture on statistics might be sort of like asking a statistician to tell you how to implant a pacemaker, but I'll do my best. So the guidance mentions what's called the MDD, minimal detectable difference. And that's a measure of the statistical power of a specific study design as performed at a specific lab. And it's actually a historical measure of statistical power for that design and that lab based on review of 15 or 20 prior studies. And it represents the threshold above which a study delta QTC will be statistically significant with 80% power. And again, it's based on historical data. In contrast, the least significant difference, or LSD, is the study-specific sensitivity, the threshold above which the study will be statistically significant, but only with 50% power. And the LSD is half the width of the confidence bounds. So if your mean effect is 5 milliseconds, your lower confidence bound is 0, your upper confidence bound is 10, the width of the confidence bounds is 10 milliseconds, and your LSD is 5. And important to note, often the E14 S7B Q&A and the training materials refer to the MDD, but they're sometimes referring actually to the study-specific variability, which is the LSD. So some best practices for in vivo cardiovascular safety studies. These are the things that you want to look for in the report. First, did they assess metabolites? You have to do that. Metabolites can be QT prolonging just as the parent may be. Second, for 5.1, the exposure should exceed the steady state high clinical exposure. How much? We'll come back to in a minute. You should be aware of the limitations of studies with very small sample size. You have to look at the study sensitivity for each study and compare that to the historical MDD. Remember, LSD is half the width of the confidence bounds. That's important because let's say the study report says the mean change in QTC was 3 milliseconds and the lower confidence interval was below 0. Statistically, that means the study did not demonstrate a statistically significant QTC effect, and you would call this a negative study. But that doesn't take into account the precision of the study. If the LSD is 10 milliseconds, which is fairly typical, that means the upper confidence bound is 13 milliseconds. And from a clinical viewpoint, the study did not exclude a threshold QTC effect. It may not be a large effect, but it hasn't excluded that. Quickly, the cardiovascular safety study report should not simply state a safety study conducted in beagle dogs, no ECG or hemodynamic changes were observed. That might be okay for the investigator's brochure. That's not okay for the report. That will not be sufficient for the regulators. Now, the time at which we are going to have to think about do we pursue a 5.1 or 6.1 scenario is typically going to be late in phase 2 or even in phase 3. And the reason for that is we need the clinical data before we can make that decision. We need to know the clinical dose and the steady state Cmax for both parent and metabolites. We have to have characterized the metabolites and human protein binding. And then we need the data to determine what is the high clinical exposure. Is there a food effect? Are there drug-drug interactions that increase exposure? What happens in the presence of hepatic or renal insufficiency? It's only once we know the clinical steady state Cmax and the high clinical exposure that we can now go and look at the in vivo cardiovascular data and the HERG study and determine if they're sufficient. So imagine it's late phase 2. We've got the human data that we need. Now we go back and look and see what sort of in vivo data do we have. And you should know what the protein binding is in the test animal species because it may not be the same as in humans. Typically, it's close for large animals that we'd be using for these studies. Next, you need to look and see not just the doses that were tested in the study, but what were the actual Cmax for the parent and human metabolites for each of the doses. And if a QTC increase is observed, the first thing you should do is see if there are any obvious alternative explanations besides the drug. For instance, if it was just due to a single outlier, or if there were huge heart rate changes or an increase in QRS that explains it. Now, if you find that there's a significant QTC effect at or below the high clinical exposure, 5.1 is not an option. And in addition, if there's a significant QTC effect at or below the clinical exposure, clinical Cmax, then 6.1 isn't an option either. So there are going to be cases where looking at the in vivo data, you realize immediately these won't support these new options. But if there are no QTC increases, or there are only QTC increases at some enormous multiple of the high clinical exposure, now you have to look at the sensitivity of the study and look at the upper and lower confidence bounds, not just the mean QTC effect. So these are data we have to look for in the reports. And do the data support 5.1? Well, first, the highest Cmax of parent and metabolites should exceed the high clinical exposure. And you also have to take into account the sensitivity of the study. If the sensitivity is excellent, LSD of 3 to 5 milliseconds, you may need to merely exclude a QTC effect at the high clinical exposure. If your sensitivity is average, your LSD is around 10 milliseconds, a negative study, that is, no statistically significant QTC effect, or negative being lower confidence bound less than 0, that's not sufficient. You also have to look at whether or not you've excluded a clinically significant effect, which is an upper confidence bound of 10. So hypothetically, if your LSD is 10 milliseconds, that's half the width of the confidence bounds. Unless your mean delta QTC is 0 or negative, the upper confidence bound cannot be less than 10 milliseconds. And if you have really kind of average sensitivity in LSD or MDD of 10 to 20 milliseconds, it's going to be very difficult to exclude an upper confidence bound of 10 milliseconds at the high clinical exposure, unless your mean QTC change from baseline is minus 20 or minus 30 milliseconds. But fortunately, high exposures cover a multiple of sins. And you can overcome the limitations of sensitivity by evaluating higher exposures. Now, the Q&A and training materials don't specifically set out numerical values, but a few guesses about what regulators would require for 5.1. As I said, if the MDD or LSD is really proof of a very sensitive study, then you might only need to exclude the QTC effect at the high clinical exposure. If the MDD is 10, you might need to reach 1.5, 2, 3-fold multiple of the high clinical exposure. Excuse me. And the higher your MDD or LSD, the larger a multiple you'll need. Again, a reminder, If you see a QTC increase, but it's at 100 times the high clinical exposure, that's not necessarily going to be a problem at all. That may be irrelevant. Now, how about 6.1 scenario? This is a little bit more complicated. First, because these drugs are too toxic to give to high exposures in humans, guess what? They're often too toxic to give to high exposures in animals. But the exposure that's tested should at least exceed the high clinical exposure. And there are statements that the study should have sensitivity to detect a QTC prolongation effect of a magnitude similar to a dedicated clinical QT studies, taking into consider interspecies differences in the normal range of values for the QTC interval. That last phrase there is actually a little bit ominous. And they give a hypothetical example similar to what I just discussed on the previous slide. There are some unresolved issues, and the biggest one I think is related to the difference in sensitivity of different species to hERG blockers. The training material states that in a dog at any given exposure, moxifloxacin results in a QTC increase that's about a third as large as in humans. So for instance, in dogs, you see a 10 millisecond double delta QTCI at 12.6 micromolar, whereas in humans, you see the same thing at about 3.6 micromolar, threefold difference. And therefore, there's an implication that if you're working in dogs or cynomonkeys, at least, you ought to be multiplying everything by 3 and using much higher exposures. And this is borne out by the other published data, which shows similar interspecies differences in sensitivity to hERG blockers, which maybe implies that instead of using the high clinical exposure, you need threefold the high clinical exposure. And instead of trying to get to twice the high clinical exposure, maybe the equivalent in the dog would be 6-fold higher. So to summarize, to support these scenarios, you need best practice hERG and in vivo cardiovascular safety study data. The study reports have to include the details that you and I are going to need to determine if they're sufficient. And we're probably not going to be able to find out if the data are sufficient until pretty late in the clinical development phase. Some caveats about what to look for in the hERG and in vivo study. And again, there are some unresolved issues. What's an adequate hERG safety margin? What steady sensitivity do we need? What about the interspecies differences in the magnitude of QTC increases? I think we're only going to get the answers to these questions as the regulators start to see these studies and learn how to interpret the non-clinical data. So I'm going to stop there and open the floor up for questions. Thanks.
I just want to say we're going to start the Q&A portion of the webinar now. We got about 5 minutes. I will invite Dr. Kleiman to put on his webcam, and I'm going to pass the controls over to Dr. Darpo and Dr. Kleiman to conduct the Q&A. I believe you have all the questions that have come in from our audience members there for you to address, and we'll try to address as many of them as we can within the 5 minutes we have. So I'll leave it to you both.
So the first question, which I'll let Dr. Darpo take the first crack at, is if you're doing an oncology study with ECGs from multiple sites, multiple different ECG machines, interpretation of QT interval by different algorithms, is there a requirement for central reading before running the concentration QTC analysis for submission?
In those scenarios, you very rarely have PK to support concentration QTC analysis, because what you're describing is a phase 2/3 study. So you would need to look at the effect using a both-time point approach, which is perfectly doable also in much smaller studies. There would be a problem, at least from some regulators, by having different ECG machines and different algorithms. So it's much more efficient to do this in a much smaller subset of cancer patients in a more stringently controlled way. So If possible, pair with PK, and if that is not possible, expand the numbers somewhat. You need to look at 35 to 40 subjects, perhaps, in a open label study, and analyze the data by time point, but then also use a central evaluation of ECGs.
And just to add a bit, more commonly, we use concentration QTC modeling for oncology studies based on the phase 1/2 first in human studies, because there we have wide range of exposures and a lot of PK data.
Yeah.
Next question I'll take is, what about metabolites in the non-clinical evaluation? What if you have 5 metabolites? Do you need to test all of those, and at what exposures? Well, I think we're going to hear more from regulators about this over the next few years. I would say you need to evaluate the exposure of the metabolite in reference to the exposure of the parent, and then decide if it's really likely to be contributing at all to any potential effect. And if it does, then in the non-clinical studies, you need to evaluate the exposures of the metabolite that you would expect to see in humans. I think we have time for one more study. Dr. Darpo, can you elaborate on whether we should implement careful ECG monitoring in the SAD, or in the SAD and the MAD, or just the MAD? As in most of our first in human studies, we get to higher doses in the SAD than we do in the MAD. So what should we be gathering data about?
My default recommendation is to implement the procedures in both SAD and MAD, because you don't know, you have a guesstimate, but you don't know for sure how much the drug accumulates, or how much the parents, how much the metabolites accumulate. So it makes sense to implement procedures. By implementing procedures, I mean making sure that the subjects are supine, making sure the protocol states that that subjects should be supinely resting for at least 10 minutes prior to each PK draw, and then having Holter's Holter recorders on the subjects, or record ECGs with a 12-lead ECG machine just prior to the PK draw. It's more efficient to use Holters, because that allows the central laboratory to extract ECGs just prior to the PK draw. But these procedures should be described, because if you end up having Holter recordings with subjects walking freely around in the site, it will come with very low quality and high variability. So you must describe in the protocol what you intend to do. Then, as I said, the most efficient approach is to just sit on the data, do not analyze, do not extract ECGs until you've seen the PK from both SAD and the MAD. Then you can decide, where did I see the highest concentrations of parent, and where did I see the highest concentration of metabolites, and therefore thereby, and based on this, make a decision on what and when to analyze.
Sonia, do we have time for one more?
Well, is it going to be a short answer?
Sure.
Go right ahead. Go right ahead. I'll wait here patiently.
The question was, can we use concentration QT data to try and waive a dedicated QT study outside of the US? Do other regulators, for instance, in Japan or other regions, accept concentration QT analysis?
Absolutely. Some of the examples I've shown has been shared also with PMDA and with EMA, and there are published examples of programs that have been approved without therapeutic study in all regulatory jurisdictions. So both Europe and PMDA is fully on board on this topic.
Wow. Well, thank you very much. I think that's all the time we have. I think we need another hour just to go through all these questions. But I just want to reassure our audience that we have reached the end of the Q&A portion of the webinar. And if we couldn't attend to your questions, the team at Clario will follow up with you after this presentation. And if you have any further questions, please utilize the email address that's on your screen, and it's heidi.mcintosh@clario.com. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated, as it will help us to improve on our further webinars. Now, give me a few seconds. I'm going to send you a link in your chat box, and you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. And get even more out of today's presentation by downloading the supporting materials that are available under the Handouts tab. And we have a copy of the slides that we've presented today, and also an update to the ICH documents are found there too. So please go ahead and check that out. Now, please join us in thanking our speakers, Dr. Kleiman and Dr. Darpo, for the very informative presentation. I hope everyone enjoyed it. Thank you, gentlemen.
Thank you so much. Take care.
Thank you.
Thanks,
Okay.
everyone.
Thank you. It has been my pleasure to be your webinar moderator on behalf of the team here at X-Talks. We thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you, everyone. Bye, Dr. Darpo. Bye, Dr. Kleiman. Bye, everyone.